Hi everybody. I'm taking up pages 324, 331, and then your protein synthesis questions. So I assigned you question 1. List and describe the three stages of transcription. So your 3 stages of transcription are initiation, elongation, and termination. Okay? And so I'm just going to refer you to this note that I drew with you guys just to save some time. So we have each of these. Initiation, we're looking for promoter. Elongation, we're going to make mRNA and make it longer. And then termination is when we get to a whole bunch of A's. Okay? And then transcription is done at that point. If the DNA template number 2 has the sequence, and that's the sequence, what would the sequence of an RNA molecule transcribed from it be? So if we look at this, we would say that we would have G, U, U, A, C, G, A, U, G, C, U, C. Okay, hope I didn't make any mistakes there. Question 3. Explain the role of each of the following in transcription. So, the role of the promoter is to tell RNA polymerase where to begin transcription. So where to begin copying the DNA and making an RNA copy. So that's the role of the promoter. What is the promoter? It's the Tata box in eukaryotes. The role of RNA polymerase is it actually makes or transcribes the mRNA. So this is responsible for making mRNA. The role of the spliceosome is that it splices out introns. So it splices out non-coding RNA. 4 says differentiate between introns and exons. So introns are non-coding sections of the mRNA molecule, and exons are coding sections of the mRNA molecule. What do I mean by that? This codes for amino acids. This codes for instructions on how to use those amino acids later. So these guys are important, okay? They often include things like promoters, etc. We're going to get to the introns later. But right now what you need to know is only the exons are kept when we're making proteins, because these guys specifically code for your codons and therefore the amino acids. Question 5 says, what are the key differences between transcription in eukaryotes and prokaryotes? So let me refer you to this bullet here. Okay, this is an excellent summary. So in eukaryotes, post-transcriptional modifications include making a 5' cap, 7 Gs are added to the 5' end, and then a string of adenines, so a poly A tail is added to the 3' end. And introns are excised by spliceosomes. So in other words, we're cutting out introns. There are no modifications to prokaryotic mRNA. So we have come up with the modifications in eukaryotes. All right, question 6 says, compare and contrast DNA replication and transcription. How are they similar and how are they different? Present your answers in table form. So I didn't give you that one, so we can move on because I thought that would be a very long question. Question 7, how is it possible for an organism to produce more proteins than it has genes for? The answer to that is you just have to think about this. Even if you just looked at the genetic code, I'm just going to grab it here, and you think, okay, what if I had, 5 leucines and 1 serine? In what order could I arrange those? And even just with those 5 of that and 1 of that, you can make quite a few different arrangements. So if you've got 22 amino acids, okay, I believe we'll only have 20 on here, but then these guys are precursors for newly discovered amino acids. If we have 20 on here, you have to think about all the ways that you can arrange it. And I'm not going to be doing the math for you right now, but the number would be huge. Okay, like you could just work with even 3 of them at the beginning and make quite a huge difference, depending if you can use 5 of these, 5 of these, 5 of these, or 6 of these, and 4 of these, and 3 of these. So in other words, the combinations of all of these guys together could be enormous. All right, so that's the answer to that question. And that takes care of page 324. And then on page 331, we have question 1. What are the key steps in the initiation of translation in eukaryotes and prokaryotes? So I'm just going to take out the note I did with you guys a few days ago. The key steps for initiation are that, this is important. I think I'm going to ask you this question again, but maybe not. Oh, an initiation. Yes, this is the question. So in initiation, you have to have your met tRNA, okay, aligned with the mRNA section that has AUG. So in other words, met tRNA has to come in contact with AUG. They're going to form those bonds because you've got hydrogen bonds because you've got the anticodon here. And then we need the small ribosomal unit and we need the large ribosomal unit to come together. Once those four things have come together, that's initiation. Okay, so that's probably the most complex part of translation right there, just assembling that unit. Okay, so that's the answer to that question. Question 2 is, what is the role of tRNA in translation? tRNA is carrying the amino acids. So it's transferring the amino acids to the mRNA, okay, and to the ribosomes themselves. Why is there not a specific tRNA molecule for each possible codon? Because this is going to be the wobble hypothesis. So I'm going to come back to this one, okay? So wobble hypothesis, important. I left this with you on your other question sheet. Question 4, list the possible anticodons for phenylalanine, alanine, and tyrosine. All right, so if we look over here, and we'll just look at phenylalanine. So sorry guys, it takes me a minute to get oriented. Phe is over here, okay? So the codons for Phe are U, 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 or U, U, C. So if those are the codons, then the anticodon would be A, or A, G. Okay? And much in the same way, we could do the same thing for alanine. So I'll help you through alanine, and then tyrosine would be exactly the same. So alanine is ALA, all right? And so our codons would be G, G, A, G, 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 U, and G, G, C. So you notice the third letter of those codons, it doesn't matter what the third letter is because it would always give us alanine. So the anticodon would be C, G, and then any of these, okay? So you'd have an anticodon of C, G, U, C, G, C, C, G, G, C, G, A. All right, and so I think you get it now. I hope you do. All right, so that was question 4. Question 5 says the wobble hypothesis states that there is an increased flexibility in base pairing at the third nucleotide of the same codons. Why does this not lead to frequent mistakes in the assembly of proteins? So I'm going to take the moment now and I'm going to explain this wobble hypothesis. So if we look at something like, let's use alanine because we've already become familiar with alanine. So alanine has 4 possible codons. Okay, your first letter always has to be G. The second letter always has to be C. But then alanine could be coded for by any one of those four nitrogenous bases. So if we look at alanine, okay, then we could have these codons: G, C, A, G, C, U, G, C, C, and G, C, G. Okay? So this is what the wobble hypothesis states. It states that if tRNA comes across a codon for alanine, okay, so this is going to be the mRNA. And this is 5' and this is 3'. Okay? So that's your mRNA molecule. And when tRNA is coming and it notices that it's carrying alanine. Okay, so for one thing, it already knows what it's carrying. I'm acting like it has a brain. It doesn't have a brain. But it knows it's carrying alanine. Okay, and so then we would have C, G, U here. All right, so this is what the wobble hypothesis states. It states that the hydrogen bonds are formed here, but not here, because it's completely unimportant what is on this nitrogenous base. Which of these nitrogenous bases is there is irrelevant because you've got all 4 possibilities coding for alanine. That's called the redundancy in the genetic code. In other words, each amino acid or this amino acid has 4 possible codons. So the last letter is of no relevance. So why is it called wobble? because these guys are forming hydrogen bonds, but these guys are not forming hydrogen bonds in theory, okay? It's a hypothesis. So the hypothesis states no hydrogen bonds are formed here. And so this kind of wobbles, all right? It doesn't form a firm bond. And so when you have an amino acid that has all four possible codons, all four possible nitrogenous bases in the third position, that's important, in the third position, the hydrogen bonds aren't formed here. And so the hypothesis states this: after seeing protein synthesis occur, we know that for these particular amino acids where the nitrogenous base, the last nitrogenous base doesn't matter, protein synthesis seems to go more quickly. So in other words, this amino acid is joined more quickly than it would be if it was for something that only has two. Or we can even look at like methionine, for instance, because that only has one codon, okay? Tryptophan only has one codon. So this would take longer because you have to form the hydrogen bond on that third base pair. This one doesn't, it wobbles and it moves on more quickly because it's redundant. It doesn't matter what it is. Okay, I hope I've done that justice. The wobble hypothesis is something I would normally emphasize on a test. There would usually be a short answer question, and I'm sure you guys know that because you would have past copies of those tests. All right. Okay, so that is question, where were we here? Question 5. So question 6 says, explain what occurs at the APE sites during the translation of an mRNA into a polypeptide. So E is for exit. The tRNA exits there. P is where the peptide bond is going to be formed, okay, between the amino acids, and A is where the amino acid enters. So that's where you get your tRNA entering, and the amino acid is now present in that ribosome. All right, question 8, using a diagram. Okay, so here I said you're just going to summarize your notes. So that is a benefit to you if you want to rewrite them and go through a process of translation. Normally you would have a protein synthesis quiz where you would draw everything out, okay? So just for your own sake, because what I'm teaching you in genetics, you are going to see in years 1 and year 2 of genetics if you decide. Like, so if you know you're truly going to be going into biology, then don't cheat yourself on what I'm giving you, okay? That's what I'm saying to you. So I would suggest doing this question, okay? Miss Delia has already said the universities are preparing for the fact that you guys aren't going to have the greatest of educations this semester. So they're already thinking about how to get you caught up. But that being said, if you don't need catching up, that's in your benefit, okay? So work through protein synthesis, know it, all right? And you know that you're going to have a quiz on it in a day, all right? And the assignment I'm giving you is extremely easy. It's just making you practice. So on that note, let us continue here. And I'm going to take this up, okay? So compare RNA and DNA, we've done that many times now. So double-stranded, single-stranded, has thymine, has uracil, stable, not stable, leaves the nucleus, stays in the nucleus, doesn't stay in the nucleus. And so now you know why mRNA, or we say RNA isn't stable, because as soon as we're done translating the mRNA, it's disassembled. Okay, so it's not used after that. Actually, it can be used over and over again, but when it's done, we take it apart. Uracil and thymine, have a look at page 314. It does give you the differences there. Okay, so I'm referring you to that page. You can see the chemical differences. Am I going to ask you about those? Absolutely not. I just like you to see them. Review the structure of tRNA. We looked at that in the note. There's a diagram of it on page 315, and there's another one, I think, on the very next page that's even more complex and better. We know that the codons consist of 3 nucleotides. How could the genetic code not use 2-letter combinations? How do we know? So in other words, why doesn't something like this code for amino acids? And that's because you wouldn't have enough codes for the number of amino acids we have, okay? So if you just had 2 letters, it wouldn't give you 20 amino acids. What's a codon? It's a series of 3 nitrogenous bases. Okay, so U is a codon. G is a codon. And then these guys code for an amino acid. Create a flowchart of the process of protein synthesis. This is just like question 8. Okay, so can you actually write protein synthesis out for yourself? That's what the question is asking you. Question 6 says, how do the stop codons act as periods? What are the stop codons? So stop codons act as periods because they signal the end of the the sentence, okay? So they signal the end of the sequence of amino acids. So that's where the polypeptide will stop. What are the stop codons? By now you should be very familiar with them. UAA, UAG, UGA. Okay, those are your stops. Which amino acids are specified by only one codon? There are two. You've seen MET. MET is super important because there's only one start. Okay, that really prevents a lot of problems. If this is the only thing that can start protein synthesis, that's a good safeguard. Okay, so we've only got one amino acid, one codon that starts protein synthesis. We also have tryptophan. Okay, so tryptophan, TRP, is the only other amino acid that has only one codon. We're going to be looking at this one in a couple days. All right. So, 8. Is the genetic code universal? What does it mean literally in terms of life and evolution? So, don't just say, okay, yes, it's universal, without really understanding what that means. The genetic code is universal. What does that mean? That means that this here, okay, if you were to find UUG in another organism, let's say a giraffe, it would code for leucine in most cases, okay? So in other words, this is the genetic code of all life in almost all cases. I can't say that. I would say maybe 98%, okay? So in other words, if you're finding amino acids, we share the same codons for amino acids in almost everything that's on Earth, okay? And the textbook even goes so far as to tell you that that's the case for many viruses as well. So what does that tell us? It tells us that we evolved from the same initial ancestor. So we taught you that in grade 11. And we've kept those genetic codes. All right, 9A, distinguish between transcription and translation. All right, important for you guys to really understand the difference here. Transcription is when we are making the mRNA. Okay, so I know I didn't write the word making there. Transcription is when you make an RNA copy of DNA. Translation is when you read the mRNA copy and assemble amino acids. Okay, to transcribe, you're going to change the language. So and I know that kind of goes against what we use the words for, but you're going to change the language. So you're going from a DNA copy to an RNA copy, specifically an mRNA copy, messenger. And in translation, you're going to read your mRNA and you're going to assemble amino acids. Question number 9, B, distinguish between initiation, elongation, and termination. So we've done that many times, okay? So please look back upon these notes. There they are for translation, you have those, and then for transcription, okay? I guess to prevent you guys from having any questions, I'll just go through it one last time. Initiation is when we look for the TATA box, all right? Elongation is when we make the mRNA. This is for transcription. And termination is when we come to a poly-A sequence, okay? And termination is done because then we transcribe a whole bunch of U's on mRNA, which just curl upon themselves later. All right. For translation, initiation occurs when you've got MET tRNA joining with the AUG codon on the mRNA. The small ribosomal unit comes together with the large ribosomal unit, and you've got those 4 things together. Elongation means we're just going to read. Okay. So that whole unit, that ribosomal unit, just shifts down one side at a time and reads and then assembles, forms peptide bonds. And termination is when we reach our stop codon. Okay, so then protein synthesis is terminated. What is a TATA box? It is a promoter. So it's a region that RNA polymerase looks for in order to know where it starts transcription. So transcription always starts after the TATA box. What is a transcription unit? I don't know if you had any difficulty finding that. A transcription unit is what we're copying. So in other words, it's everything after the TATA box all the way till we get to those A's, okay? So it's whatever is being copied in protein synthesis. What is the coding strand? So when we have our DNA molecule, DNA, okay, not RNA, so we're in the nucleus and we're beginning transcription. This is the template strand. And this is the coding strand. Okay, so the template strand is what we read. The coding strand will look almost identical to the mRNA, and that's why it's called the coding strand, but you don't use this one, okay? Never use that one. It'll look almost identical to the RNA, except for the fact that you're going to have T's on this and U's on your mRNA. So we use the template strand, we don't use the coding strand. Does only one RNA polymerase work at a time? This is important because I left this for this point here, not to overwhelm you earlier. Absolutely not. So as soon as RNA polymerase is working along this template strand and assembling an mRNA molecule, another one could be up here starting. As soon as this RNA polymerase, let's say it's down here assembling, another one could come in here and start assembling. So you're going to make many, many, many RNAs, okay, mRNAs. So absolutely not only one is working at one time. It wouldn't be efficient to undo those hydrogen bonds to only make one. We make many of them. So if we need a protein, we don't just need one protein. We're going to make many proteins, okay? All right, what is the Wobble hypothesis? I've gone over that with you now. And what is charging tRNA? I wanted you to be just familiar with the term. Charging tRNA is this. When the tRNA leaves the exit site, okay, so whatever it might have here, AAA, okay, when it leaves the exit site, it goes and picks up another amino acid. And just to be completely accurate, think for a second, which amino acid is that going to be? Okay, while I find it on here. And it's going to be phenylalanine. Okay, so charging tRNA means getting the amino acid back onto the tRNA. That's what it literally means. Okay, and how did I get AAA? I had to look up the opposite. So I looked up U, and it's phenylalanine. Because remember, this is the anticodon. The true codon is U. And so that's what you'd look up. All right, good luck with everything.